I suppose I'd quite like to ask a question, because obviously there's a clear difference between PhD student and then someone who's been doing science for decades. Kind of what got you into looking at the nitrogen cycle, looking at nitrous oxide emissions?
He's the nitrous oxide guru.
Yeah.
Okay.
Yeah.
I'm a charlatan here.
Okay.
Well, I did my PhD on carbon dioxide release from forest ecosystems.
Okay.
So nothing to do with agriculture.
No.
But when a job opportunity came up, I was finishing my PhD off, and they wanted someone who had really good, they wanted expertise in greenhouse gas emissions. They wanted someone who had expertise in organic manures. And I was on the periphery of both, working in forest ecosystems and on CO2. So I asked my boss what he could do, and he was just pulling together a new mobile lab. This was Phil Einison in York, the first mobile lab for measuring greenhouse gas emissions in the field.
Oh, wow.
Okay, a bit like what Daniel uses on now. And he said, come with me, come downstairs. And we went and had a look outside. He says, have you ever used a GC before? I said, no. He said, you need to put that on your CV, don't you? I said, yeah. He said, just push that button there. So I pushed the button. He says, well, you can now say you've operated a GC. And then he said, we'll come back later. We came back later, and these were the days before all the digital outputs. Everything got printed out.
Oh, so it was like
a
pen
big
on a
pile
paper.
of, yeah. So he said, just bring that pile up to the office. And I shared an office with him at the time. He says, you can now say you've handled data. And so we wrote
physically as well, rather than it being
exactly. So we wrote it, and I just got into. I was lucky. I got to the interview, and they said they wanted me. And the project was looking at how livestock manures impact on greenhouse gas emissions from soil.
You've stayed in that though, so there must have been something in there that kept pulling at you, or is it just again circumstance?
The nitrogen cycle is unbelievably leaky. So you can make a career out of just trying to understand the nitrogen cycle and trying to improve it. And to be honest with you, that kind of work, the manures, the nutrients, quite applied work, really gets under the skin after a while. And I really, really enjoyed it. Really enjoyed it. I still do. Yeah.
So in terms of experience with N2O, because I have a trailer as well that has automated systems. What's your experience with automated systems, long-term measurements, N2O?
Do you want to answer that one? Or? I mean, I can't. But.
Well, yeah, I have some experience with long-term systems. So I've got a similar story to Dave about how I got into this, which was that I worked at the International Rice Research Institute for years, rice paddies, which produce a lot of methane, and were blamed for a third of the global methane sort of 20 years ago, based on figures in a test tube from some lab somewhere. And we got money to set up these chambers in paddies, fixed chambers for measuring methane and then later nitrous oxide. And we had 8 of these, 5 countries, 8 different stations with this elaborate closed chamber system and GCs in a porta cabin to automate it all. And we showed that methane emissions more like maybe tops 10%, more like 5% paddy methane emissions of the global source. So the poor rice farmer was not going to be numbered for reducing methane emissions. So that was a while ago, and then an opportunity came back when I was back here. One afternoon I was told, you've got an opportunity to apply for $500,000 to get some bucks for a kit. So I dashed off a proposal which was somewhat like these methane boxes, but to look at CO2 and nitrous emissions from grassland with isotope measurements to separate the sources of the different gases. And to my amazement, we got it.
It's Wilson.
That was the picture of Wilson.
Yeah.
And then I luckily got people who knew how to actually make this stuff, and we've got a pretty tasty lab out of it.
Yeah, that's what Chris is working on.
Cool, But it's kind of happenstance that we ended up getting this thing.
Yeah. Well, I got involved with automated systems first of all in the lab. So the denitrification system that Laura works on, I remember interviewing Laura, and that was the project that we were going to work on. So it's very much about trying to develop a system that could really look at the fundamental processes that drive N2O and N2 emissions and be able to measure those simultaneously, which of course is very difficult to do because the atmosphere is full of N2. So that was the first part of it. And then when I was still at Northwick, we developed a much more applied system that we could use outdoors to measure from livestock manures and from urine patches, etc. So They're really good. They're great because they give you that high resolution data, which I'm sure you know about, Sam. And there's so much. They're expensive and they can be a bit temperamental. But when you look at the opposite, the other end, the cheaper end of the spectrum, where we're using static chambers, which we've used for, decades. They are so labour intensive. Taking the sample from the head space, taking several of them so you can measure that concentration increase, bringing them back to the lab, those samples that then have to go through a GC. If you want to have lots of replicates and lots of treatments, you're probably better off using the static chambers.
Yeah.
But if you want high resolution data, to really understand what might be driving some of these emissions, then the automated system's really quite good. So, you've had a few issues with yours, though, haven't you?
I've had a few issues.
So
yeah.
what system have you got?
So at the moment, I'm running some Picaros. They analyze N2O, CH4, and CO2 connected to some Lycos, doing the automated bit of moving the chambers. But they generate a lot of data. So is it something you've worked with, a lot of data like that? Because it's measuring the chambers like every hour, so you have a lot of data points.
Yeah.
But
does it measure dinitrogen, or is that only in the dentist? I suppose with the flow through.
Yeah.
I think that's something I recently did my first experiment in the dentist system, that incubation system that was set up at Northwick. And unfortunately, although I didn't get very good nitrogen data because I used a different type of core that had a closed bottom, because my bulk density was quite low and I didn't want soil to fall through into the system. But the idea, I love the idea of being able to measure nitrogen, because it's not very well understood how much nitrous oxide gets then converted to dinitrogen, which of course is there's so much of it in the atmosphere. It doesn't matter when it gets converted to nitrogen, compared to nitrous oxide being a very strong greenhouse gas and ozone depleter. And I think that's also could be considerable. I think one of the big benefits of
it's absolutely right. If you want a career, if you want to denitrification is probably total denitrification, N2 emissions. It's one of the most understudied processes in the N cycle, because it's so difficult to measure. We've used the acetylene inhibition technique for years, which stops that nitrous oxide reductase, so nothing gets converted from N2O to N2. So you count all your denitrification fluxes as N2O, and you have cores with acetylene and cores without, and you can work out what the N2 emission is. But there's all sorts of caveats around that. Tried using it in the field, diffusing acetylene into the soil underneath static chambers. We've tried that as well. But trying to measure dinitrogen, as you might imagine, is not easy. So there are people now using, as you're probably aware, 15N techniques to be able to give you that dinitrogen emission.
Do you work with modelers to unscramble this data?
So CEH has a lot of modelers, so I've been asking their help with the data, because a lot of it is just continuous data. So you have a massive spreadsheet, and I found out that Excel has a limitations on number of lines you can have of data. So working with
how
modelers,
frequently are you collecting data from the headspace then?
It's measuring a core every hour. So there's 16 of them. So every hour it just goes around them.
I understand that, but I guess it's a continuous flow going into the analyzer, is it? And so the analyzer is measuring the concentration. How frequently is it measuring the concentration? Every second?
Every few seconds. I think it's every second, so it's just pulling it continuously.
Wow.
Yeah, so you could reduce that and still get a fantastic relationship in the cumulative emission over time, a cumulative headspace over time.
Yeah.
Because you're probably getting, how long is it closed for?
It's closed for about 2 minutes.
Oh, only 2 minutes?
2 minutes,
okay, that's not so bad then. Sorry, I didn't realise that.
So it's 2 minutes every hour for per
that's probably
core, and then repeats all 16.
Yeah.
It equilibrates rapidly enough in 2 minutes, is it?
Yeah, it does, yeah.
Oh, it's such a sensitive instrument.
It's very sensitive. If you leave it for longer than 2 minutes, you end up...
Does it start to saturate?
It saturates.
Does it?
How large is the chamber?
I'm using the LICO auto chambers. They're quite small.
The sort of white CO2 ones.
Right.
So if you leave it for longer than 2 minutes, you have problems.
And I thought I had a lot of data, because the dentist system does one every 8 minutes. And
I have too much data. I've had to ask. So it's a good thing that I have a lot of modelers around me and people who understand R and how.
I was going to say using R.
Yeah, using R. So
if you've got data, you're in a position of power. Modelers are out there craving data, and if you pick and choose your partner.
Yeah.
Yeah, absolutely. But you've got aspirations to use another kind of equipment to measure the greenhouse gases, haven't you?
Yeah, so this one, because what I have at the moment is just measuring soil, but I want to see what happens when you have soil and plants, and not just small plants, large plants. So say if you have wheat, so we have something called Skyline, which is like a massive chamber that will be able to just enclose a whole plant or a couple plants, and then you'll be able to analyze what's happening in there. So it will still be using the Picaros to analyze the gas, but
Yeah, same analyzer, but
same
different.
analyzer.
Have you seen the Skylines?
I have, yes, yeah. Actually,
Yeah.
Sam showed me and Ben them when we were down there.
Yeah.
Yeah, they're great, aren't they? Oh, it's a great concept anyway. Just needing one chamber, it just lifts up,
Just make it
comes back
bigger.
down.
Just make
Yeah.
it bigger. yeah.
And how big is that chamber?
It's 60 centimeters high. I can't remember the width now, but it's quite wide. It's probably this wide. But I've got
And it can be any height you want, can't it? Because
It can be.
you can make it the sort of Tedlar bag type approach. Rather than having a fixed wall,
Right.
you actually use a Tedlar. I think it's what you use, isn't it? A Tedlar
Yeah.
kind of bag that comes down
it's
over the
somewhere-
top. So you could have it over birch trees or something like that. Miscanthus. I've seen people use it over miscanthus before.
Mm.
But you wouldn't get a rapid enough change in the nitrous concentration to detect, would you, anything that volume?
You probably have to play around with the length of time you leave it closed. But if you
Yeah.
use something like the Picaro or the quantum cascade laser type approach, they're so sensitive they can pick up differences in not like a GC.
Yeah,
You can really pick up differences in the headspace.
Yes, yes, yes. Well,
concentration.
we have a Picaro. And
Oh,
we've
you
got
do?
and a Picaro isotope analyzer. We started out with a mass spec, which was a nightmare in the porter cabin.
Oh, yeah.
They're like British motorbikes, you know, they've got an oil leak.
That's what I'm
You've got to treat them as a lot of TLC. So we struggle with that, but the Picaro is so much more robust. It's not quite as sensitive, but you can throw it around. And so, yeah.
Yeah, okay. So what kind of treatments are you guys
I
looking at?
do you want me to go first, Sam? or would
You
like
can
to?
go first.
So I'm looking at simply at the moment changes in water concentration, but over time. So a lot of people who study the nitrogen cycle kind of say okay, the conditions are this at this very moment in time, and that's why we're seeing this certain peak. But there's this effect that's been observed wherein the historic moisture conditions, so the soil's legacy, if you will, or I think Dan likes to call it, was it the land's memory, or
Oh, yes, he did
something
something today,
poetic
didn't he?
along
Yeah.
those lines. But basically, if the soil's been dry for a little bit before it's wetted, then it seems to produce a much bigger peak in emissions
Of N2O.
of N2O.
Yeah.
And even if it's wetted to the same kind of water-filled pore space as a wet sample that's then wetted up to that same water-filled pore space. So if, for example, if you to keep a sample at 50% and take it to 90%, and keep a sample at 70% and take it to 90%, the sample that's gone from 50 to 90 will produce a much bigger peak in N2O emissions. So my first experience was just simply altering the water. I didn't add any fertilizer to it. I didn't add any glucose to it. I just changed the water, and there was something like an 8-fold increase in the dry treatment compared to the water treatment, compared to the wetter treatment. Yeah.
So I'm looking at two treatments, plus the control, of course. So I'm looking at water table. So raising the water table from, normally they use about minus 50 centimeters at the farm where I took the soil. And so using that.
So they manage the water table
at minus 50 centimeter water
for
table.
what?
So
What
they're
kind of-
peat soils. They're trying
Ah.
to preserve them. And so the way they preserve them, they raise the water table, especially in winter, and so when they don't have machinery going on it. So they keep it 50, anyway, they claim. And then we are proposing you need to be higher, between 20 and 30. So I'm looking at...
In order to do what? Why would you suggest to keep it higher?
So when you lower the water table, there's a lot of oxidation of the peat, so it burns away, it just wastes away.
So you lose a lot of CO2.
Less CO2.
Yeah.
So, but if you raise the water table, it means you still have the top 20 centimeters, which will be affected, or 50 centimeters if you use the traditional. But hopefully you have fewer emissions. So we're looking at that to see the N2 emissions from that. And then also we're looking at another mitigation measure, which is adding crop residue, fresh organic matter. So in the experiment I'm doing at the moment, I've added barley straw. So just mixed in some barley. And so I've got treatments looking at that. So just comparing which is most effective, just
Yeah.
water table alone or water table with fresh organic matter, or just leave it as it is.
So that's a really important point you just made then, that you're not just measuring nitrous oxide, oxygen carbon dioxide is the one that really you could imagine. That's what they're trying to prevent, that loss of CO2, loss of carbon from these soils by keeping them anaerobic. But you're looking at the interaction with the other greenhouse gases, because you might increase N2O because you're making things a bit more anaerobic. So you're looking at the whole greenhouse gas balance.
Yeah.
And methane.
And methane.
Yeah, and
So,
methane,
yes.
yeah. Okay, yeah, important, absolutely.
Yeah.
With yours, with you just describing this sort of big peak in N2O, you get, are you looking at the CO2 and methane as well?
So I looked at CO2 and there was absolutely no difference between the samples, which I thought was really weird. I would expect one of them, because the microbes might be getting very excited, which is why you might see this N2O thing. No, the CO2 levels are very low. And in terms of methane, although we did collect data for methane, we didn't calibrate it for it. We didn't have a standard for it. I haven't or won't be able to interpret that.
Really? Can you not just put a methane standard through the GC now? Because the FIDs are quite linear normally. Well, it'd be better than nothing.
Yes, yeah, you're right.
Yeah.
You're right. And I personally, I think, and I didn't. The problem is I didn't think about it enough, and I didn't realise that the Dennis system could also do methane. So when I got the data, I was like, oh, there's this methane thing, and maybe I should have, because a complete greenhouse gas balance would be wonderful.
Yeah.
Just looking at kind of the raw peak data, there doesn't seem to be be a difference.
Yeah.
There
Peak
doesn't
areas.
seem to be just peak areas.
Yeah, okay.
There doesn't seem to be a difference. But it'd be good to actually put some concentration to that and see whether there is a difference.
Chris mentioned today that he was doing CO2, but this next season he's going to be adding fertiliser and nitrogen. Is he going to be doing N2O then? Have you got the facility to do N2O?
Well, we do. So we've got the Picaro analyzer. We have a continuous flow-through system with this isotope analyzer in line.
Right.
So we need to take it out and substitute for the
Oh, I see.
the nitrous analyzer. And the plan is to do that for short intervals following fertilisation intermittently. But the main purpose is to have this continuous trace of CO2 and its isotope signature.
Yeah, great.
Mm-hmm.